All right, good afternoon, everyone. I'd like to welcome you back. We're reconvening for our afternoon set of talks for our 16th International Conference on Invert and Fish Cell Culture. Just to remind you, we're convening this. My name is Vivian Day. We have had Cindy Goodman and Shirley Pomponi, also my co-conveners, and we're going to have again another round of talks that are for the afternoon. More about current and future applications, but also wanting to highlight the wonderful sponsorships that we've had for both the morning and the afternoon sessions. So once again, thank you very much to Schmidt Ocean Institute, Cryocrate, and SIVB for sponsoring this one-day conference. For my little few-minute introduction, I thought when we're talking about the current and future applications of invert and fish cell culture systems, when I was pondering how to sort of go about introducing this afternoon session, I actually thought about how most of us are thinking about the future and how AI may impact us down the road. So I was curious to see if I typed in current and future applications of invert and fish cell culture into an AI image generator, what would I see? So this is what I got. A bunch of, and for those of you that maybe are towards the back, might be a little bit challenging to see this, but essentially it's a bunch of sort of bell jars, very futuristic looking fish. Not as many inverts, I do have to admit, even though the word was in the AI prompt. But in the end, when you look at it, is this our future in terms of invert and fish cell culture systems? So if you really want to zoom in, this is what the AI image generator actually gave me. So in those little jars, it was not a fish that looked like a fish. It was a fish with a bunch of tails and whatnot. So, thinking about the future in terms of AI, and I thought, well, maybe it was just that AI image generator that was kind of giving me those odd sort of illustrations. So again, popped it into a different, the same prompt, just sort of into a different AI generator website, and I finally was able to get something that resembled some inverts. Now, that being said, we have some amazing speakers lined up for today to take us from our background that we talked about earlier on in the morning sessions and seeing where are we essentially going. So we will have these talks both for fish and a variety of different inverts, but I do want to also just highlight once again that no matter if you're presenting or not during this afternoon session, you are more than welcome to submit a paper for our special issue for the In Vitro Development Journal. So we are putting together a special issue on current advances in invert and fish cell culture systems. If you do happen to want to take a snapshot of this QR code, it'll take you to our submission page. And remember, the submission deadline might be September 30th, but that being said, as papers are coming in, they are being reviewed and are then going to be published online, pre-print. So definitely don't delay to get that in. So without further ado, again, you can see this in our schedule that we do have a number of wonderful talks set up, but I want to make sure that I give Dr. Lucy Lee, our SIVB Lifetime Achievement Award winner for 2024, her fully allotted time to take us all through fish in vitromatics and in vitro omics as our first session. So with that, I'd like to introduce Dr. Lee, who is just relatively recently retired as Dean of the Faculty of Science at the University of Fraser Valley in British Columbia, Canada. So, Lucy, please take it away.
Okay, there we go. Can you hear me now better? Okay, great. So this is going to be my talk. I have collaborated with my husband and former PhD supervisor over the 35 years that I've known him. And so we collaborated in fish cell cultures. So that's what I'm going to be talking about. And these are two terms of several that we have coined. And you are all, by the way, in vitromaticists. Okay? But in vitromatics and in vitromics is something that I want to talk a little bit about. And what it is that in vitromatics really is the history and the methodology for doing cell culture. So you have to go back always to the history of it. And this one, I know you cannot read it, but it gives the highlight. And so I figure I'll write it a little bit so that you can see that. And now I think you can read this. You all know cells were described 350 years ago, and more actually. Scientists culture cells outside an organism for over 100 years now. And continuous cultures, or cell lines, have really only been in existence about 80 plus years. And it begins in a way, not with HeLa, but a mouse cell line, the L cells. And there is a lot of derivatives of the L cell line now. It was established in 1943. And you all heard about HeLa cell line, Henrietta Lacks. That was published in 1952. It was established earlier from the cervical cancer of Henrietta Lacks. And it's been going around the world, and now there is tons of that cell line. And Henrietta, of course, passed away shortly after when they had taken the biopsy. Another point that I want to emphasize is that 1955, Eagle was one of the first ones to define nutritional needs for growing cell lines. And I say surviving in vitro because both my husband and I are from that era, from that decade. Now, 1960s and 70s, this is the golden years of cell culture. And I emphasize more now for the fish people. The first fish cell line, RTG2, was established in 62. So did the insect cell lines, actually, was published in 62. Serum-free containing culture media was first pioneered by Richard Ham, but there were others working along that. And then the discovery of growth factors. I was hoping to see Dan Risato here because it was his father's work, but I don't see him. 1980s, there was a lot more going on. In the 2000s, there's, of course, quite a lot, but I'm emphasizing what I want to talk about, the new applications of fish cells. And this is why I am busier than ever, because my work up to 2017, it was okay, no big deal. But then all of a sudden, my phone, my email, my contact is ringing crazily. So this is where the first fish in vitro meat startup started. I was their founding advisor. And then I moved on, of course, and they moved on. And the other thing is that RTGIL-W1, which is a cell line that my husband and I published in 1994, now is a cell line that is actually used by the Organization for Economic Cooperation and Development as a test guideline, and it is also an international standard, an ISO. So let's go and see. Oh, the reason I also wanted to point this is that this person, and I can't pronounce her last name, Magdalena, had a nice setup in there, but also emphasizes Atala. Atala demonstrates 3D and organs bioprinting techniques. So Atala is a MD, PhD, who is known for doing bioprinting for medical applications. And they are far ahead than the cell culture meat, that they are printing just a slab of meat. Atala's group has been printing organs. Atala, Tony Atala, was invited to SIVV in 2017. And I became buddies with him. Well, not quite, but we chatted a fair bit. And Tony is my age and also from Peru. He is a fellow Latino. I look Korean, but, and I am Korean, but I grew up, my formative years were in Peru. So Tony and I chatted quite a bit that day. And so I want to start with how we develop cell lines. The reason why I'm getting an award is because I just culture cells, and I was able to develop a lot of cell lines. And so developing continuous cell lines, which is the history and science of establishing, characterizing, storing, and distributing cell lines, is not just a science, it's an art. There's a lot of tricks of the trade that you need to know, and Anita has, a lot of those. And also, you gotta have those green fingers. Some people grow plants really well and some don't. Well, those green fingers, Nathan over there, he has them. Okay, so you can do, in a way, two ways of growing cells. You can do an explant method, and, oops. Can you play this? Like, it is a video. Oh, go back. Click on the image, and it should work. Yeah. So the explant method, and if you don't want that, you can also do a QR code, and you have a different one in here. This one is a new cell line that we haven't published yet that the explant is growing. This is time-lapse, so you can see the cells growing out. They form a monolayer, and then you neglect them for a while, and then they start growing. So the other way is doing enzymatic dissociation, in which you take a tissue, you chop it up, and you get it into single cells using various enzymes. Mainly trypsin has been a favorite one. And then you can place in culture, and hopefully they'll grow. So going back to first fish cell line. The first fish cell line, RTG2, was published in 1962 by virologists, because that was the application for cell lines, to grow viruses, because viruses do not grow on their own. They need host cells. And for the human impetus was also virology. So was all these other vertebrate and invertebrate cell lines in a way. But the biggest impetus was to be able to grow viruses. And I love the way they used to write in the past. This was a publication in Science. These two lines is the full abstract of that paper. Isn't that beautiful? Anyways, nowadays, there is about 1,000 fish cell lines. And there is a database that collects those as much as feasible. And I was in a meeting with them in Switzerland in January. And they keep this database, and it's updated all the time. So the first time when I used it was in 20. But in 22, I cataloged it, and I had a nice bar, just the blue bars in the background. And then I went back just last week, and I wanted to see how many cells of the different species there are. These are the top 40% of all the reported fish cell lines, but there is over 20,000 fish species, and it's only like not even 0.001% of the fish cell lines available. And most of the fish cell lines that are available are from rainbow trout. Then zebrafish was fourth in 2022. Now it's actually second with goldfish, and so forth. And there is a whole bunch of other, these are the top numbers. And then there's a whole bunch of cell lines of different species, but not that many compared to human. There's over 100,000 cell lines of human, whereas only about 1,000 for the fish. So if you want to know about all the different cell lines and what they do, you can check Cellosaurus. That's the one to check. Now, these are some of my published fish cell lines. And as I said, people kind of ignore me until, the fish meat industry, in vitro meat started being hot, and then they realized they need a fish cell cultures. And there aren't that many there. Now there are a few more. And there's a lot of mammalian cell cultures who jump into the thing thinking that they can grow fish cells, and then they have problems, and then they're calling me. Anyways, my first cell line was RTLW1. I published it in 1993. But I started this, and I know exactly the date, August 9, 1983. That's 41 years ago. It took me 10 years to publish that one. And the subsequent ones have been a little faster to the most recent one that was published with Georgina Dowd. She's a scientist in New Zealand. I went in 2018, and we got it published by 2022. So 4 years, so that's good. But it does take a long, every one of these cell lines, and these are the published ones. And then I have in my nitrogen dewar, have over 50 different fish cell lines in there. So, and RTLW1 I will talk about, and RTGIL2. The rest, maybe I might be able to touch a bit, but just want to emphasize that this cell line is not the last one. This is another one that we just submitted. And the chief editor of In Vitro Animal just told us we need to do a little revision, but hopefully it gets accepted soon. This is the isolation and characterization of 2 cell lines from the gills of this, which I can never pronounce, Chrysophorus auratus, or Australasian snapper, and Oncorhynchus chawisha, which is Chinook salmon. I was invited by Georgina, actually in 2017 I went there too, and we started doing the cell culture. So the 2017 one, I think, is this one, and the 2018 was the other one that we published. So anyways, we're working on this, and hopefully we get it published so you have access to these new cell lines from the gills. In the meantime, I love trout. And I have dissected the trout inside out. If you want to know the anatomy of any fish, you can ask me, and that's one of the reasons why they also call me a lot, because I actually have a long time training with just the anatomy, comparative anatomy of different animals. And if you know the anatomy of the fish, I have every cell line possible from every tissue, including some obscure tissues, for example, the corpuscles of Stanius. I don't know if you have heard of it. I have also a cell line like that. My more recent ones is RT-olf, the olfactory tissue. I discovered that you can grow cells from the olfactory tissues of any fish, 100%. I have tried at least 20 different fish, and I have cells from those. The mesothelium, RT-meso, also we have cells. We have cells from the tail muscle, and that's the one I'm going to talk about. We also have heart cells. We have kidney cells. We have ovarian fluid cells, which Christine Schirmer just asked me to send them to Switzerland. So I just did before coming here. Can you play this one, please? These are rainbow trout ovarian fluid in the first year. Up to about 5 passages, we had the cilia from the ovarian fluid. And then eventually they disappear. But we have been characterizing it, and I'm slow at this, and I'm sorry, Nguyen, that it's taking me forever to publish, but we'll get there. But this is immunofluorescence of the epithelial cells. They're beautiful epithelial cells. They express zonal occludens 4, and Nguyen is very good with immunofluorescence techniques. Okay. So, fish cell lines. They have a lot of uses. This one is a slide that I borrowed from my husband because he likes to think of all the little details, which I kind of have a tendency of bypassing because I go at 50 miles per hour, whereas he goes at 10 miles per hour. But that's okay. We work together. And there is this many, if not more, applications. And I don't and a lot of the times I've given a lot of talks about for the study of viruses or the study of bacteria, because there's a lot of bacteria that also affect fish. But now biotechnology is the biggest application. And then and I'll give you an example of microbial disease. Toxicology is big with Christine Schirmer, who is leading the use of this RT-GIL-W1 as a standard for in vitro toxicity testing. And so many other areas that I would like to touch, but we have no time. So I will start with RT-GIL-W1. That was the second cell line that we have published. It was a student. Oh, I don't have the pictures. Oh, that's too bad. I think it's in my other talk because I'm giving a talk tomorrow as well. I'm giving 2 talks. But anyways, Christine, although was not an author in the paper because Christine started writing to Nels that she wanted to do graduate studies, and he was sort of ignoring her. But I said, you better take that girl. She's very good. And now she's like the director of this center in EOWAG, and she has her own company as well. And she and her group have been able to make it so that it is an ISO standard and a guideline from the European Organization for Economic Cooperation and Development. Yeah, I always get that wrong. So anyways, this is the beginnings of what we call the in vitromics, the uses or applications of cell lines. And what we have is that every day, there is about 15,000 new substances that are registered with the Chemical Abstract Service. And of the 100+ million chemicals that have been registered so far, very few are actually regulated, and even fewer are assessed for their toxicity, for their safety. So the OECD, the Organization for Economic Cooperation and Development, has standards or guidelines for that toxicity testing. And their main one was this before, the OECD 203 fish testing that requires at least 5 tests to be tested, plus 1 control, so that's 6 tanks with 7 fish per tank, because you gotta have the replicability. So that's at minimum 42 fish per compound. Well, if you use the cell line, you can use it over and over and over and over, and it's only 1 fish. You can also use RT-GIL-W1 to study intracellular pathogens, pathogens that grow inside and are difficult to culture. And so this was my student before, Rochelle Monahan, who came as an undergrad, and actually most of my students have been as undergrads. Nathan started as an undergrad as well. Rochelle is now the vice dean of the Faculty of Science, so I'm so pleased to see how my students have come so far. Could you play this? Is it working? Yeah, I think it's working. No? Yeah. So, because I culture cells and all this, sometimes you see some oddballs, and sometimes because you get it from, and you may end up having some intracellular parasites that you did not know about. Although it wasn't from my own cells, we noticed that a cell line that was shipped from China, one of those fish cell lines, had something like this. And I thought, that's odd, like I don't have no idea. I'm not a parasitologist, but then I had to study parasitology just to understand this. These are called the microsporidians. They are intracellular pathogens. They affect some insects, and that's how I got involved with over there, that I wanted to study Apis mellifera. What was the microsporidia that affects? There is several anyways. But anyways, Ancalia algera is another microsporidia. It doesn't normally affect fish, but it affects humans as well as, well, its main host is mosquitoes. And there has been cases of zoonotic transmission of Ancalia algera infection in humans from people just swapping these mosquitoes. Anyways, we wanted to grow this now in culture and observe them. And so Richelle had the patience to look in the microscope and be able to record it. And you can see these guys are really weird. They have their nuclear material inside, and then they eject it out. And so it sends its cytoplasmic contents into another cell, and then it divides in there. It's an incredible mode of development. I also used RT-GIL W1 to study some parasites, external parasites, and even I looked at big parasites. But this one was one of the first ones in which my students, then she's a teacher and she's an MD, PhD, who worked with me to study this bug called Neopermiba pemacuidensis that causes gill disease in fish. We also have a collection of cells, which we call the in vitrome. And the fish in vitrome, as I said, it's about 1,000 fish cell lines. But I'm going to emphasize now in the fish muscle in vitrome, that is cell lines of fish muscle. And that, in a way, started with this paper in 2014. And there's Nathan there. Very good. He's actually in several of my papers. And then he's an independent researcher, of course. Sarah, Nathan, Andrew. Andrew was from UFE. These two were from Laurier. And Michael, also from Laurier. But they are all PhDs, and he's an MD. So my students have been done really well. And Sarah and Nathan helped me derive this cell line, which is really the first myogenic. It actually shows muscle fiber striations, which I have only seen in Michael's. Where's Michael? There, right? Yours have those striations. Okay. Also, we did several other cell lines that are sort of myofibroblastic. They do have a bit of the muscle markers, like, say, for example, alpha smooth muscle. Actin, vimentin, collagen. S100 is just a marker for neural crest derivatives. But desmin is one that should be in actual muscle, but it was negative. And these are beautiful pictures. Yes? 5 minutes. Oh, I'm running out of time. Okay, so this is one of the other, the last one that we just got published, which is a myofibroblast precursor cell line from the tail muscle of Australasian snapper. Okay, rainbow trout mussel cell line. This is our newest one. We haven't published it, but we're giving it away because I'm retired. I don't want to work with it. It does show some striations. I don't know if you see that when it peels off. These are some of the markers for mussel lineage anyways, and we've only done MyoD, and that's what it's showing, but we still have to do all these other ones. A recent paper in 2022 just got published by Tsuruwaka and Shimada. I don't know if they're here, and they have used, I'm not sure, their fish, I think it was a snap or something, but anyway, this just came out. This looks exactly like my mussel cells when they peel off. They do form lipid droplets, and mine that do too, and I don't have the picture. And they do form these little organoid type things, and it's exactly the same. So I got really excited, but I didn't have the time to take the pictures. Anyways, I have other cells that accumulate lipids. These are the mesocells, same with oil red O. This is the KFE cells, and you can see the banding striation and collagen forming cells, and they have several of those. This is one, the goldfish skin cell line, which was published in 1997, that it was done, I guess this is an older version, because I did have a picture of them. Anyways, right now, I also have a cell line that has been transfected with GFP. But the beauty of these cells is that they have been transfected over 15 years ago, and they continue, and they are really green, and you pellet them, and they're green almost. So they're beautiful. They are monocytes. This is their normal morphology, kidney shape, and then they are phagocytic, and when they phagocyte things, they kind of lose their the GFP, but we do have the fluorescent beads in there. Anyways, I have done work in Chile, Norway, New Zealand, Japan, and hopefully soon to Korea. And then the Chilean sea bass was one that we were really excited because they grow at 4. They don't like any other temperature above 4. So you have to keep them in the cold. And I don't know if they survive, but when I was there, we were growing them in the fridge. Okay, so I think I'm going to end here because this is something that the next ones are going to talk. But I do want to emphasize that I have met Florian and Maria Worm, and they are the sort of founders of XL Gene. And they are a biopharmaceutical company. And they have told me that CHO cells, this is the Chinese hamster ovary cells, they have capable to produce over 3,600 tons of CHO cells per year. That will make the in vitro meat a reality. And since inception of the bioreactor technology, over 2.5 million metric tons of CHO cell biomass has been created. Isn't that amazing? Anyways, I'll end it there. I'll take questions.
Questions? We have 5 minutes.
Lucy, if you compared a given toxin in same number of cells and same kind of medium, with or without serum, in fish cells versus a mammalian cell, what would you see in difference in terms of specificity, species specificity?
For the serum, or?
No, for the toxin in a fish cell versus 3T3 cells or keratinocytes or something else.
Yeah, well, first of all fish cells are very slow metabolically.
Yeah.
So they would take at least 3 days to double, whereas mammalian cells will double every 24 hours. You know, like CHO cells are capable of doubling even under 24 hours. Yeah. The other thing is that they made, for example, CHO cells are adhesion dependent, so they need a a surface to adhere. But then they developed the technology that they, much like a previous speaker talked about, making them to grow in suspension. And so now, all this for biopharmaceutical production is done in suspension. And we need to make fish cells to grow in suspension. The only one right now that I have are the blood cell lineages, so RTS11, and then we also have RT kidneys that are also in suspension. But we do have, we were able to make ASG10, the Atlantic salmon gill cell line, under different conditions to be in suspension, and it's really exciting. But those are gill cells. I do have the muscle cells sort of like forming like kind of organoid types, but we're still far behind the mammalian technology. Yes?
Thank you. Kind of building on the toxicity question, since you've looked at so many different vertebrates, so many different cell lines from those same vertebrates, do you notice any big trends in sensitivity to toxins? Like, are you finding that like certain cell types are overall more sensitive?
Yeah, but the other thing is the mode of delivery of the chemicals and the medium in which it is used to deliver to the cells. Some cells are more sensitive than others. And Anita this morning already talked that, for example, for rotenone, the sensitivity between Atlantic salmon gill cells and rainbow trout gill cells were different. You could see that difference. And there is a lot of people that have published that in the literature and also comparing with mammalian cells. And there is differences, yeah. Guy.
Guy?
Thank you. I love a lot fish. I have been eating insects, but I also like a lot fish. I'm impressed about these numbers. These are really high numbers in terms of production. But I have a question. I will continue with the question that I also asked before. In terms of, let's say, omega-3 fatty acids, are these cell lines producing it de novo, or you need to provide, let's say, precursor molecules in your culture medium to have the production?
Yeah, you have to provide precursor molecules, which are already in the culture.
It's no criticism, eh? It's just
Yeah,
to know how the system works.
And because fish prefer linolenic acid over linoleic acid.
Yeah.
And so they do make de novo, and that's one of the big differences between mammalian cell membrane and fish cell membrane. The composition is different, yes.
Yeah, it's like with insect cultures. They also cannot make
Hi, Lucy.
Hello.
So many cell lines that you have created over your rich career. Which one is your proudest?
I'm the proudest. I would say my first one.
RTL?
RTLW1. They are 41 years old and they are still growing. And what I did is that I refused to submit it to the American Type Culture Collection because in terms of citations, RTLW1 is giving me more citations than RTGILW1, which was submitted to ATCC. So everybody just cites ATCC and not Lee et al.
Thank you.
Okay, with that, thank you again, Lucy. I'm sorry. Sorry, just a very quick practical.
Do you distribute your cell lines? Okay, you're handling all of that.
And right now I'm giving them away for free. Everybody's getting it. Well, I am charging for the cost of the materials.
Okay, thank you again.